Hey everyone, and welcome to the Explainer. You know, right now as we're speaking, inside every single one of your cells, there's this incredible process happening that's turning in invisible code into, well, you. So today, we're going to crack that code.
I mean, really think about that for a second. Your DNA is the master blueprint, right? It's this long sequence of letters, tucked away and hidden. So how on earth does that blueprint get turned into things you can actually see, like the color of your eyes, or things you can't see, but are absolutely keeping you alive right now?
Well, the answer is a process called protein synthesis. It's basically the cell's way of reading that DNA blueprint and building the actual stuff of life. And the name makes perfect sense. Synthesis just means to make something. So we are literally going to explore how your cells make proteins. So why are we so focused on proteins? What's the big deal? Well, it's because your DNA doesn't directly make your hair brown or your eyes blue. Instead, DNA holds the instructions for making proteins, and it's those proteins that do all the heavy lifting.
And that's not an exaggeration at all. Proteins are the absolute workhorses of your cells. They're transporters carrying things around, they provide structure like the scaffolding of a building, and they act as enzymes, these tiny little biological machines that speed up all the chemical reactions your body needs to survive. Without them, nothing gets done.
Okay, but we have a problem, a logistical one. In our cells, the master blueprint, that precious DNA, is locked up tight inside the nucleus for safekeeping. But the factories where proteins are actually built, they're outside the nucleus, floating around in the cytoplasm. So how do we get the instructions from the vault to the factory floor? We need a messenger.
And that messenger's name is RNA, or ribonucleic acid. RNA is just perfectly designed for this job. You see, unlike the super important double-stranded DNA master blueprint, RNA is just a single-stranded, disposable copy. It's got a slightly different sugar, and it swaps out one of the DNA letters, thymine, for something called uracil. So you can think of DNA as the priceless original cookbook, while RNA is like a quick, temporary photocopy of just one recipe that you can take into the kitchen.
So this whole process of making that RNA copy, it's got a name, transcription. And the script part of the word is a fantastic clue. The cell is literally transcribing or writing down the message from the DNA language into the RNA language. And this all happens safely inside the nucleus.
So how does this actually work? It's really a beautiful three-step process. First, an enzyme called RNA polymerase comes in and unzips a small section of the DNA. Simple enough? Second, that same enzyme zips right along one of the DNA strands, using it as a template to build a matching strand of what we call messenger RNA, or mRNA. Finally, once that message is fully copied, the new mRNA molecule just peels off and heads out of the nucleus, ready for the next phase of its mission.
Okay, awesome. Our messenger has the code, and it's made it out into the cytoplasm. But before we can actually build anything, we have to know how to read the message. And it's not as simple as reading one letter at a time. The genetic code is actually read in 3-letter words. And these words are called codons. Each little 3-base codon on that mRNA molecule corresponds to one specific amino acid. And those amino acids, those are the building blocks we're going to use to assemble our protein.
Now, to translate these codons, scientists use a really cool tool called a codon chart, and I'm going to walk you through exactly how it works. Let's say our codon is AUG. The very first step is to find that first letter, A, over on the left-hand side of the chart. See that? That narrows it down to one entire row.
Okay, step 2, we find our second letter, which is U, along the very top of the chart. Now, you just follow that column down until it meets the row we already found, and just like that, we've narrowed it down to a single box. And for the final step, we just find our third base, G, on the right side of the chart. Follow that line across, and bingo, we land on our answer. AUG codes for the amino acid methionine. And here's a fun fact for you. Methionine also acts as the start codon, which is the signal to begin building pretty much every protein.
All right, your turn. Let's do another one. What if the mRNA codon is CCA? Go ahead and try it. You'd find the first base C on the left, then the second base C on the top, and finally the third base A on the right. And what do you get? Proline. See? It's a really simple but powerful system for decoding the language of life.
So, we have our message, we know how to read it. What's next? Well, now it's time for the main event, actually building the protein. And this all happens in the second big step, which is called translation.
Translation is a team sport, and there are three key players. First, you've got our mRNA, which is the message itself. Then you've got the ribosome, which you can think of as the factory or the construction site where the protein gets built. And it's actually made of another type of RNA called ribosomal RNA. And finally, you have transfer RNA, or tRNA. These are the transporters. Their whole job is to go out, grab the correct amino acid, and bring it to the factory.
So at its heart, translation is all about converting from one language to another. We're translating the language of nucleic acids, those codons on the mRNA, into the language of proteins, which is just a chain of amino acids. This is where the code becomes a real physical thing.
And this happens in 3 phases. First up is initiation. The mRNA latches onto a ribosome, and the very first tRNA shows up, carrying methionine, and it locks onto that start codon. Next comes elongation. This is the main part. The ribosome just chugs along the mRNA, reading each codon one by one. And for each codon, the right tRNA molecule shows up, drops off its amino acid, and they get linked together into a long, growing chain. Finally, we hit termination. The ribosome gets to a stop codon, which is exactly what it sounds like. It's the signal to end the job, and the brand new protein chain is released to go do its work. So, we've gone all the way from a code locked in the nucleus to a finished protein floating in the cell. When you put all of that together, you get one of the most fundamental concepts in all of biology. And this is it. It's called the central dogma of molecular biology. The flow of genetic information in pretty much all life on Earth goes in one direction, from the master blueprint, DNA, to a temporary message, RNA, to the final functional product, the protein. That's it. Transcription, then translation. So, a great way to think about it is that DNA is the brilliant architect who designs the entire building. It holds the master plan, right? But it absolutely can't do the building itself. It relies on its dedicated crew of RNA molecules, the MSNN courier, the RNA construction site, and the tRNA delivery trucks, to actually get the job done.
This entire process is just mind-bogglingly precise. But what happens if there's a mistake? A single typo in that original DNA code? Well, that one tiny change can alter a codon, which could bring in the wrong amino acid, which could cause the final protein to get misfolded and not work at all. And that's actually the basis for so many genetic diseases. It just shows you how incredibly critical this whole process is for our health. It's an amazing system and one that is so worth exploring more.